Sulfur, the nutrient that acts like the nuts and bolts that hold all of core plant functions together. Today, we're going to be talking about sulfur, what it does in the plant, how it's taken up by the plant at the root zone, and when it's in the soil, the limitations and antagonisms that can cause sulfur to not be taken up by the plant. So a plant builds a bunch of proteins in order to perform essential functions. So when a plant is making a protein, it takes a bunch of these amino acids, and sulfur is a key component in most of them. So cysteine is an essential amino acid, and when it creates proteins, it breaks apart, and this sulfur forms with other cysteine amino acids and creates what's called a disulfide bond. We can think about this disulfide bond like the rivets or the cables on a bridge. They don't necessarily make the bridge itself, but what they do is when that bridge undergoes stress, like heavy winds or a heavy load, they make sure that it doesn't fall apart. That's the exact same thing that these disulfide bonds that come from those cysteine amino acids do for proteins. That structure could potentially get denatured if it falls apart. This denaturing happens through heat, oxidative stress, or salt stress, and a host of other problems that could stress a protein or stress your plant. The plant is creating these proteins. They need the disulfide bond at all of the corners to hold that protein together, especially under stress. The sulfur prevents that protein from immediately just falling apart. But the protein doesn't necessarily need the sulfur in order to be created. It just helps a ton with preventing stress. So it's really interesting. When we don't have enough sulfur, then our protein folding doesn't stay exactly how we want it, and then those core functions stop happening, and your efficiency of your crop to take light, turn it into sugars, all of that drops because you don't have the key proteins to maintain efficiency across all of those essential plant functions. So while sulfur doesn't necessarily do one thing, it does everything. And it holds everything together in a specific way. So a super interesting fact about sulfur in wheat plants. In a wheat plant, you have the seeds that contain protein. Now the plant made this really interesting method of determining which proteins to fill that seed with. It generally has high sulfur proteins and low sulfur proteins. The high sulfur proteins are more resistant to denaturing and have downstream effects. When that wheat gets turned into flour and gets turned into dough, it's way more elastic and stretchy and is good for bakers and can hold and trap air correctly. The plant will tell itself which proteins to create, either high sulfur wheat seeds or low sulfur wheat seeds. And it does that through epigenetics, turning on or off certain genes that promote either the high sulfur protein or the low sulfur protein. And if we have a deficiency in our plant, then that plant will turn on the low sulfur protein, which is not good for our quality. That's just one example of how sulfur, while not necessarily decreasing yield, can decrease quality if it's not there in all of the proteins in your plant. Plants take up sulfur in the ionic form of sulfate, SO4 2-. Sulfate is chemically inert and it needs what's called an activation. This activation step turns this sulfate ion into what we talked about earlier, that cysteine amino acid, or one of the host of amino acids used to create these proteins. That activation requires 8 steps. A lot of those steps even require sulfur in order to turn sulfur into a usable form. Once it's in that usable form, it can be translocated through the phloem, but only after it's activated and only a little bit. That means it's not necessarily mobile or immobile. This sulfate ion is very mobile. It goes up through the xylem, but it's immediately grabbed, activated, and turned into an amino acid, decreases in its mobility because it's often locked into a protein. It can't unlock itself and then go back up the plant like some other nutrients can. It's not necessarily immobile either because the entire amino acid can sometimes move up the phloem and around the plant. That's why you see sulfur deficiencies differently than you see an immobile micronutrient or a mobile macronutrient. Sulfur is kind of in the middle. Sometimes you see it, halfway up the plant or like sometimes right below the new leaves. Things like that, which makes it a really interesting deficiency to look at and catch. Now you may look at this sulfate ion and you notice that It's an anion. It has a negative charge. Something else has a negative charge. Nitrate. When we think of nitrate, we often think of nitrate leaching or nitrate leaving the soil because it doesn't have a cation to grab onto our soil particles. That's the same thing that happens with sulfate in our soil. So it's something that we got to pay attention to is sulfate leaching. We have less and less sulfate if you're not adding back to it what the crop's taking out. And oftentimes we're adding, sulfur to the soil indirectly when we're adding things like gypsum or other sulfate containing nutrients and fertilizers. Although if you're not adding those nutrients, then your sulfur is going to just be decreasing as the seasons go on. Sulfur is generally found in organic matter. That's why most sulfur deficiency symptoms happen in sandy soils with low organic matter that are heavily irrigated. And so sulfur can leach really easily. So it's important that we monitor the amount of sulfur that's in our soil. Generally that's through organic matter, and then monitoring throughout the season in tissue tests to make sure that we're getting sulfur to the plant. Sulfur is generally found in soil in an organic form, meaning it's already in a protein that's in the soil, but it needs to be mineralized by a microbe in order to turn back into sulfate, which can be taken up by the plant. But that sulfate ion, that inorganic form, that's easily leached. So that means as soon as that microbe turns it into a sulfate ion from the organic matter, or the sulfate ion comes in through gypsum, and as soon as the water hits it, the calcium separates from the sulfate. That sulfate can easily be leached if it doesn't have the microbial community to grab onto it and turn it into an organic form of sulfur. So it's really important to be watching those levels because they can leach in one season. You might have heard of competition between nutrients at the root uptake sites with things like calcium, magnesium, potassium. If you think of Mulder's chart, like how different nutrients can antagonize each other and prevent uptake, sulfur only has one nutrient that it can prevent uptake with, and that's molybdenum. If you look at both of those nutrients in their ionic form, they look the same. So both of them have this O42-, meaning they have the potential to compete with each other at the root zone because there are specific transport proteins at the roots that use this 2- charge to pull nutrients in. That means if you're growing, say, a legume that needs more molybdenum to nodulate the roots and have that symbiotic nitrogen uptake, that if you're applying heavy amounts of sulfur, say a soybean crop that's in a sandy area with low organic matter, you may prevent any amount of molybdenum from being taken up due to this competition dynamic. So we want to be watching our molybdenum levels in our tissue tests if we're applying heavy amounts of sulfur directly at the root zone. Now to recap, sulfur acts as the nuts and bolts to the proteins that your plants build to do essential core functions. They prevent the proteins from collapsing on themselves, from denaturing under stress, under heat, salt, anything that can stress a plant. Sulfur does this by going from a sulfate ion through a whole 8-step process of activation to become a cysteine molecule or any other amino acid that forms the building block of all of the proteins in your plants. So it's really important, but it's super interesting because it doesn't do anything specific. It just holds all of your proteins together. It's also really important in photosynthesis in what's called a ferredoxin molecule, which is iron and sulfur together to help take the energy from the sun and turn it into sugars. It's just a helper nutrient all throughout your plant. Now in the plant, it's not mobile, but it's not immobile. It's kind of in the middle. It's in this gray area. It's mobile in its sulfate form, but it's immobile as soon as it gets activated, which is pretty immediate. And then after that activation, when it's in a cysteine molecule, before it gets turned into a protein, then it can be remobilized and moved through the phloem. Sulfur is easily leachable. So the sandy, low organic matter, high irrigation areas are going to be most at risk for sulfur leaching and are areas to be aware of and to look for any kind of deficiencies. I'm Jack. I'm the founder and CEO of Cultra, a nutrient analytics platform for crop consultants. And today we were talking about sulfur. You're welcome to go to cultra.us, take your soil tests and your tissue tests, drag and drop them into our site, and you can see where your levels are and have it interpreted for you. And you can see if your sulfur levels are getting leached season by season or if they're increasing. And then you can overlay on your tissue tests and see, is that sulfur actually getting into your plant? Hope to see you there. Thank you so much for watching this video, and have a good one.